This section explains how organoids derive themselves and why that self-derivation carries all the way through to their applications. An organoid grows itself. Give a starting cell population, the morphogenes it lacks, and a scaffold that permits 3 dimensions, and the tissue runs its own developmental program to completion. First symmetry breaking, then patterning into spatially distinct domains, then morphogenetic rearrangement into organ-like architecture. The chapter's whole move demotes the engineer from constructor to condition setter. You supply only what the endogenous program cannot generate itself, and self-organization carries the rest. The mouse optic cut needs almost nothing beyond a low-factor medium. The stomach derived from human pluripotent stem cells needs staged exogenous cues into foregut fatant stem cells needs staged exogenous cues into foregut fate. Everything downstream rides on one consequence. Development studies, disease models, drug screens, personalized medicine, all of it follows from this. The same self-patterning that builds the organ reproduces the pathology whenever the seed genome already carries the lesion. This section explains why the lung is the hardest organ to study and how growing a substitute in the lab turns that difficulty into a general method. The lung resists study because it is both inaccessible and irreducibly plural. It holds 58 cell types spread across epithelial, endothelial, stromal, and immune categories arranged in a branched architecture that no biopsy samples whole. The chapter's move is to stop reaching for the organ and grow a surrogate instead. You seed stem cells, supply the developmental signals, and let them self-organize into a mini lung in a dish, one that recapitulates the real tissue's cell-to-cell structure and function. The generative primitive here, the one you can carry to every later chapter, is substrate substitution, where an organ is unreachable, an organoid stands in, because the same developmental program that built it once will rebuild it in vitro. The lung is merely the hardest case, the one that makes the payoff of the substitution legible. This section explains why the respiratory chapter reaches for a lung organoid instead of a conventional model. A lung is about 40 cell types folded into a branching gas exchange architecture. A flat monolayer or a mouse cannot hold that complexity, so every model built by construction loses the very thing being studied. The chapter's move is to stop building the model and grow it instead. Give stem or progenitor cells the right microenvironment, the matrix, the growth factor cocktail, and the geometry to sense it. Then they rerun the organ's own developmental patterning in miniature, self-assembling the cell diversity no one could assemble by hand. The fidelity is inherited from that developmental program itself. That is what earns the phrase innovative technology. The disease model and the patient-derived biobank come free once self-organization does the work. Within the book's arc, this is the organoid as platform claim, specialized to the respiratory system. This section explains how a brain organoid is grown. A brain region is specified. You hand pluripotent cells the right morphogen gradient, the SHH, WNT, and BNP axes that set dorsal ventral and rostral caudal position in the embryo, and the tissue self-organizes into that region on its own. An organoid is developmental biology, rerun in a dish. You supply positional signals early, then withdraw them and let intrinsic cell fate programs finish the sculpting. Directed differentiation paints the coordinate deliberately. Undirected differentiation lets the cells find it on their own. Either way, you buy a human 3-dimensional model, cortical, cerebral, midbrain, or forebrain that two-dimensional culture and mouse brains cannot give. The catch the chapter flags is this: a mini-brain that works too well raises bioethical and bioplausibility questions. This section explains why brain organoids are the right instrument for studying neurodegeneration. Neurodegeneration resists study because the object is inaccessible. Human brain tissue is ethically locked. Animal brains are the wrong species, and flat culture strips away everything that makes a brain. A brain, one cell type, no architecture, no cell type. The chapter's move is to make the model isomorphic to the modeled rather than a proxy for it. A pluripotent stem cell, given only the developing brain's own morphogen cues, that is, WNT and SMAD inhibition, self-organizes into a three-dimensional cerebral organoid. That organoid reconstitutes cellular diversity, cytoarchitecture, and, past roughly 120 days, cytoarchitecture and, past roughly 120 days, validity is bought by recapitulation, not by similarity. And because the starting cell can be the patient's own induced pluripotent stem cell, the same instrument becomes personalized medicine. This section explains why the reproductive system needs organoids and what shape that answer takes. Flatten a reproductive tissue onto a dish and it forgets what it is. Two-dimensional culture strips away the cell-to-cell contact and the matrix that hold a gonad's phenotype, so the model drifts from the organ it was meant to be. The organoid is the correction, a three-dimensional niche where cells reorganize and recapitulate their behavior. In vivo. This chapter is the book's universal move applied to reproduction, forked by source. Adult stem cells can be drawn directly from ovary or testis, or pluripotent cells can be differentiated toward a reproductive fate. The reproductive specific payoff is this: patient-derived cancer organoids become personal biobanks, each one a private testbed for drug screening. The organ whose telos is already generation, now modeled by a structure that self-organizes toward it. This part explains why packing mesenchymal stem cells into a spheroid switches on cartilage and bone formation. The starting idea is that condensation itself is the instruction. Cartilage and bone begin in the embryo when scattered mesenchyme packs into dense aggregates. The crowding itself, not an added factor, licenses the chondrogenic and osteogenic programs. A monolayer never crowds. It spreads, stiffens against gigapascal plastic, and stalls. Force the same mesenchymal stem cells into a non-adherent drop, and they recondense. The plasticity modulus collapses toward the kilopascal softness of living tissue, and the developmental switch throws. Hundreds of hypoxia, angiogenesis, and differentiation genes shift, and the chondrogenic and osteogenic yield rises. The spheroid recapitulates ontogeny, so it stops being a cell suspension and becomes a building block, a preloaded morphogenetic module you either transplant directly or tile into macro tissue. This section explains why biomaterials are the second input organoid development needs. A tissue's final shape is underdetermined by its cells alone. The surrounding matrix supplies the missing boundary condition, the constraint that fixes what the genome leaves open. Biomaterials are that second input, the extrinsic channel through which you steer the self-organization. The genome alone will not resolve. They are how you reach in from outside and shape the tissue. Madrigal is the default, and it wins on biological richness. It drives proliferation and spontaneous folding. But it is a found material, ill-defined variable from lot to lot, animal-derived and untunable. So it stimulates growth, yet cannot direct morphogenesis. It gives you abundance without steering. The field's move is from found to design. Synthetic hydrogels, such as PEG and hyaluronan, whose stiffness and chemistry can be dialed toward a target architecture. You set the parameters instead of inheriting them. The governing axis is biological richness versus parametric control. Every new scaffold is a position along that axis, and clinical use pushes hard toward the defined, controllable end. This section explains how genome editing turns an organoid from a passive model into a causal instrument. Give an organoid a programmable write head and it stops being a passive model and becomes a causal instrument. CRISPR. Cas9 edits one disease gene, the organoid regrows its phenotype, and you read the path from gene to disease directly. Correlation is upgraded to causation, and the same rig doubles as a therapy test bench. The chapter is one move, catalogued across organ systems, the neurosphere, the hematopoietic, the retinal, and so on. Every entry runs the same loop. Knock out or restore a gene, then watch the sick phenotype shift. What gates the method is not biology, but two unsolved engineering problems: Cas9's off-target cuts and delivering the guide RNA into the target cells. Fix those two, and the loop generalizes to any single gene or multi-gene disease. This part explains why an organoid's usefulness and its ethical danger are the same property. The same fidelity that makes an organoid useful makes it a moral problem. That is the whole chapter compressed. Every enumerated concern, cell source, donor consent, legal status, chimaera, gene editing, transplantation, biobanking, misuse is one axis of a single question: how closely the construct resembles. Self-organization is morally neutral until it succeeds too well. A brain organoid or a gastroloid provokes artificial life and humanization arguments precisely because the developmental mimicry worked. So ethical liability and translational value are not two things to trade off against each other. They are the same variable. Read twice. Predict the next case from this. The closer a construct climbs toward the real organ, the sharper every concern on the list becomes, all of them at once.